Fantastic. Hi there. So welcome to this biology paper 2 walkthrough. My name's Stephen. I will be doing the live stream for you today. So we're going to be looking at the A-A paper. So all these questions that we'll be doing will be coming from the A-A Excel board. That doesn't mean if you're doing PR, Excel or anything else, you can't follow along. I've purposely chosen questions that contain content that is relevant to all of the exam boards. So the content in any of these questions could come up in your exam. And additionally, we're going to be focusing here on exam technique. So we're going to be looking at the types of words that you'll find in exam questions, the types of questions you'll find, and they are common to all different types of exam boards. So no need to worry about that. We've got a chat running, so if you would like to put a comment or a question or anything you would like to put through, feel free to leave it in the chat below. I believe you just need to subscribe to the channel and you'll be able to put a message into the chat as well. If you are after any further questions after this or anything else, you can find myself on Sherpa in the box below. So do have a look at that if you need to. Fantastic. So what we'll be doing is having a look at some various different exam questions and exam techniques. What we'll just look at first is kind of what we'll see as we go throughout the session today. So this is our kind of holy grail of top exam tips that we'll be focusing on. So the first one here is understand the command words present within your questions. Okay, so these are things like describe, explain, evaluate, and all the different command words you might be asked to do for a question. And we'll look at what each of those means and what we actually need to get out of it. Again, be aware of how many marks are actually on offer in the exam question. If, for example, it's a 3-mark question, we need to be making at least 3 specific points. If it's a 5-mark question, we need to up that to 5. Okay, so we're just focusing on how many marks are there and what we can put down because of that. Questions are not trying to trick you. Think about what you have learned and apply it to the question. Now, let's be very superficial. What I mean by that is that everything that you have learned can come up in an exam. They will not ask you something you do not know. They will present you with new information and expect you to apply what you know to that question. They're never trying to trick you out on something that you've never heard of before. Okay? They will always give you context to your question. And we'll see some examples of that. Now, of course, specifically biology, it's not just about content, practical skills and skills here at GCSE. So we're going to focus a few questions looking at this, so that we're not just solely focused on content. And as well, running on the time, it's very commonplace for you to be told by your teachers all the time. But let's make sure we're not losing because we don't finish the paper. Now, you can revise as much as you can. One thing I absolutely love is that exam technique can really, really help. It's all very much cramming as much revision as you can to this last week before an exam. But looking at exam technique could actually help you score you more marks in these last few days before an exam because you'll be in the right mindset to tackle the questions, whatever the content might be. So that's the generic outline of what we're going to look at. So let's crack on with a few exam questions and put all of this into practice. So the question that we're going to do here today is going to be based on some context recall and high-level maths question to get us going. So our first question here, if you can see this on the whiteboard, my advice would be to follow along through the question and see how you go. Jot down any answers you have on a spare piece of paper or on a Word doc and follow along as you see how we answer the questions. Fantastic. So the first question here, energy flows through an ecosystem and materials are recycled. Figure 6 shows the water cycle. So below here, we've got in Figure 6 an unlabeled diagram of the water cycle. Nice and simple first question, name process X. So we can see on the diagram here, we've got X labeled here, and the question is asking you simply to name that process. It's 1 mark, we just need to write one word down, and that's all we need to do. So for this one here, we've got what looks like a pond or a lake, and it's showing the sun that is taking the water from that lake and converting it into what looks like water vapor in the atmosphere that's then going to condense and form clouds that will then precipitate down as rain. So what is the name for a process that takes water and turns it into water vapor? Well, that would simply be evaporation. Okay, so a simple 1 mark question to get it started. Nothing too bad, but again, it involves looking at the diagram and taking your knowledge that you've learned throughout the course and applying it to the question. The second one down here says, name the process by which water is absorbed into plant roots. Now, this is a bit of a tricky one. It's not actually related to the topic that the previous question is related. In other words, we're not looking for precipitation or condensation or anything like that. It's asking the name of the process by which water is absorbed into plant roots. Now, if we know anything about water, we know that, second, just have a look at this. Right, so good. Yes, let me just go and grab some cookies just to make the audio a little more Right then, is that any better for anyone? Let me know in the comments. Does that seem to come across a little better? Not as crackly, hopefully. Yes, do let me know in those comments if that is a little better for you there. Yeah, any better there? Little, hopefully it's a little easier to hear me with the headphones in. Fingers crossed that's the case. Okay, then, well, I haven't seen anything else on the chat, so hopefully that is a little better. So I will carry on for now. But if that is a problem, do just let me know in the chat. Fantastic, awesome. So as I was saying, we're now moving on to a different topic. It's water. If we ever think of water, the most immediate thing that should come to mind is osmosis. So osmosis is the way that water is transported into and out of cells. And that's the same for plant root cells, okay? So although it's not directly related to evaporation, it's still kind of linked because we're talking about water. So don't get tricked out there that it's suddenly talked about something different, but we're using a bit of information from another part of the course. That's all good.
And here 6.3 gives 2 uses of water in plants. So here again, just recall. Remember, the most obvious one is photosynthesis. So you can write down things you remember. So photosynthesis is carbon dioxide plus water. To remember this one, glucose and oxygen. And so we can see here water is used in photosynthesis. So that can be one of our answers here, photosynthesis. But equally, we can mention other things like support. Water is really important in terms of the turgor pressure within the cell. Remember that vacuole within the cell can take up water. So we could say it's useful for support. But there are loads of things we can say. It's a solvent. So minerals and ions, sugars can dissolve in that water to be transported through the phloem or the xylem. And it's also useful for opening the stomata, various different things within the plant. So there's lots of answers here. We just need short, we don't need lengthy sentences here. 2 uses of water, just short bullet points. Fantastic. Right, so moving to a slightly trickier question then. So here we've got a question that goes on about food chains. And we have a very scary looking 5-mark maths question that we're going to tackle. So figure 7 here shows the flow of energy through a food chain. The numbers are in kilojoules per meter squared per year. So here we can see that sunlight is coming into the food chain, and the grass, or a primary producer, is using that sunlight for growth. Now, some of that energy is being lost to decomposers or other herbivores that might eat it. But whatever energy that plant takes on, it can pass up the food chain to the next organism, in this case a cow, and that will take in the energy and use it for growth, again giving some away as respiration, some away as feces. This could quite easily be an exam question in itself. Why is not all of the energy converted to growth? Well, it's lost through metabolic processes like respiration or through feces. Over on the right here, the cow is more efficient than the grass at converting energy. The energy conversion efficiency of the cow is 4.098%. Calculate how many times more efficient the cow is at converting energy than the grass. Now, very helpfully, it gives you an equation. So the equation for energy conversion is energy conversion efficiency equals energy used for growth over energy input times by 100. So what we can do to work out this question? Well, even though it's 5 marks, we don't need to be super scared of it. 5 marks just means they are picky in the mark scheme and that they just want to see your working out. Now that's important for every single maths question on this paper. So what we can do is we can see that they've already calculated it for the cow. That's 4.098. So what we need to do is calculate it for the grass. So what we can do is look at the grass first, and we can just plot numbers into this equation. So here we've got energy used for growth. So first of all, we can take 21,800 because that is the energy here that is being used for growth by the plant, and divide that by the energy input, which is 1,700,000. And then we can times all of that by 100 to give us a value of 1.282. So we can see that our value for the grass is 1.282 compared to 4.098 for the cow. Now, just to check that, we can do it for the cow just to make sure. So we can say for the cow, the amount of energy used in growth is 125. The energy input is 3,050, and we can times that by 100, and you will get an answer of 4.098. So you can actually use what they've given you to work out whether you've done it correctly yourself. Now, unfortunately, that's not the end of the question. We're asked to calculate how many times more efficient the cow is at converting energy than the grass. So don't just leave the question here. Go back and make sure you've answered exactly what it's asking of you. So what we can do for that is we can take our value for the cow, so 4.098, and divide it by our value for the grass, which we've calculated as 1.282. And that gives us a value of 3.196. Okay, fantastic. That is not still the end of the answer, because it asks you to give your answer to 3 significant figures. Now, what that means is that you need to have 3 figures here instead of 4, which means you need to round up your answer. So our answer to 3 significant figures is 3.20. 3 significant figures, a 3, a 2, and a 0. Okay, so zeros are significant. So we could put in there 3.20. Okay, so a tricky question. It seems like it's got so much behind it because of the 5 marks. But as long as you put down your working out, you're getting marks here for taking information from the question and putting it into the equation, getting an answer out of that, doing the correct divisions here, rounding your answer to 3 significant figures. There's lots coming out of it. So just be sure to make sure to put down all of your workings out. Any questions, again, just leave them in that chat, okay? So moving on, just to finish off this question, we have 2 little shorter answers here. So 6.5 says it is more energy efficient to rear cows indoors than to rear cows outdoors. Simply give 2 reasons why. Now, this just asks you to think back to content from course. So we do a lot of information about intensive farming and good farming techniques. So why is it better to rear them inside compared to outside? Even if you have no clue, just think of the differences between inside and outside. Inside warmer, okay? So they're not going to lose as much heat. So less energy lost as heat. So that could be one point you make. Additionally, they're indoors, they're kind of kept within smaller limits, they can't move about as much, so they're going to lose less energy from movement as well. Okay, so we're really thinking about the words that question is asking us. And 6.6 suggests 2 possible disadvantages of rearing cows indoors. Again, this is a type of question, but it's simply recall from the course. Okay, they're not giving you any extra information. They're asking you to just recall what you've been taught. So here, think again, why is it not good if they're all hunched together? Well, you could say anything from you've got an increased chance of disease. So if they're all packed really tightly together, that disease can more easily spread throughout the population of cows. As well, when you use them indoors, you'll learn in the course that you can use more antibiotics. So that would be one. If you're giving them lots of feed, that feed will have antibiotics in. It's a big problem, rather than using grass, for example, if they were reared outdoors. You can also put more superficial things. For example, it would cause them more emotional stress, or the welfare of the animal would be less inside. Equally, you're more likely to get aggressive behavior indoors because they aren't in their natural environment. And any of those would be equally as fine to write down. Awesome. So a question where we're looking at some tricky maths content, but also some recall as well. So let's move on to some slightly different questions. Now, one thing that always comes up every single year is genetics, no matter what exam board you sit. So we're going to look at one of these genetics questions where you have to write out a Punnett square and tackle that, because that is always one that comes up and is very different to any other question you find on the paper. So let's have a look at this. So here we've got maple syrup urine disease, or MSUD, is a rare inherited human condition. MSUD is usually diagnosed early in childhood and can be controlled by having a low protein diet. Figure 4 shows the inheritance of MSUD in one family. So here we've got a family tree. Now it gives us a key, so we're going to just spend a bit of time before we answer this question understanding the key. So we've got males as squares, females as circles, and any shaded circle is an individual that is affected by MSUD, and any unshaded circle is an individual that is not affected by MSUD. Now, a key bit of information we are told down here, the allele for MSUD is recessive. Now, remember what we mean by that term recessive. It means we must have 2 copies of the allele to be affected, okay? In other words, any of these individuals that are shaded here, they need 2 copies of that recessive allele to be affected. Remember, you have one allele from your mother, one allele from your father, and they can be different. In this case, if they're both recessive, you will be affected. Give one piece of evidence from Figure 4 which shows that MSUD is a recessive condition. Now, again, this question's come up a lot of times. It's a common one that they use. We can tell here because we have offspring from parents who are not affected. So what that means is that these individuals up top here, 1 and 2, they can be carriers. Now remember, carriers are where you carry an allele for the disease, but are not affected yourself because you have a dominant one. So let's designate a capital N to be the dominant allele and a lowercase n to be the recessive allele. So if we've got 2 parents here, we've got a capital N and a little n. If they are carriers, they carry this recessive allele, which means if they come together and produce offspring, then the offspring could potentially have 2 recessive alleles without the parent being affected. Okay, so our answer here is simply that parents without MSUD have offspring with MSUD. Fantastic. Right, so now the tricky part. Now let's have a look at the Punnett square itself. So here we've got the diagram remade down the bottom here. And what this question is asking us is persons 7 and 8 in figure 4 are expecting a 4th child. So we've got persons 7 and persons 8. Determine the probability that the child will have MSUD. You should draw a Punnett square diagram, identify the phenotype of each offspring genotype, and use the symbols N for allele for not having MSUD and little n for the allele for MSUD. So what we need to do is draw a Punnett square, and we also need to answer all of these different bits that it's telling us to, okay? So we're not just going to ignore what we should do, we're going to do exactly what the question says. So Punnett squares, we know these. We're going to draw out Punnett square. They look like this, a little box with 4 quadrants that we can draw out like so. The parent's genotype. So this is really kind of the crux of the question, the hardest part to understand. If the parents are unaffected, that means that they can't have 2 recessive alleles. So their genotype is not going to be NN. It's not possible because otherwise they would be affected. They'd be shaded in. So the question is their genotype homozygous dominant, in other words, capital N, or heterozygous, capital N, little n? Well, if they are having offspring that are affected, as in person number 12, they must carry a recessive allele somewhere. And both of them must do that in order to get 2 recessive alleles in person 12. So we know that the parents, both 7 and 8, must be heterozygous. So we know that for sure. So what we can do is we can write into our Punnett square that. So first parent here, we're going to write out their genotype, capital N, little n, and then in the second, for person number 8, the same, capital N, little n. We can then cross through our Punnett square. So N combined with N is homozygous dominant. Big N, little n. Big N, because we're taking it from up here, and little n, because we're taking it from over there, and then combining the last 2 for 2 little n's. So we've drawn a Punnett square. Fantastic. We're now asked to identify the phenotype of each offspring genotype. Okay, so remember the genotype is the genetic characteristics that make up an organism. So this is the N or little n. The phenotype is the physical appearance of that genotype. Okay, so in this case, it's do they have the disease or do they not? So we can write out for N, capital N, that this individual here, they are going to be unaffected. Okay, so we can just write that they're unaffected, don't have NSUD. We've got two individuals or two possibilities of being heterozygous. Again, they've got a dominant allele covering up the effect of the recessive allele, so they will also be unaffected. And because you need two copies of the recessive allele to be affected, the little n will be affected, so they will have NSUD. Okay. All that's left to do then is just put out probability that the child will have NSUD. So we can see here that we've got 4 options, so we can just divide 100 by 4 to get 25. We've got a 25% chance that they are homozygous dominant. We've got a 50% chance that they're heterozygous, because we've got 2 heterozygotes here, and then a 25% chance that they're homozygous recessive and therefore will be affected. So we can then write in that there is a 25% chance or a 0.25 probability. And you can see down here at the bottom of the question, it just asks for the probability. All you have to do right there is either write in 25% or 0.25. Okay, and that will be 4 marks for that question. So a pretty tricky one. Again, they will give you information you've never heard of. In this case, NSUD, this interesting disease. We do know how to read these family trees. We do know how to construct Punnett squares, and we can get an answer out of that. Fantastic. Right. The question then goes on. So this question then goes on to ask some tricky content for the higher tier part of the paper. And this is where all of that content comes from. It's all the higher tier stuff in the second part of the question. So figure 5 shows chemical reactions involved in the normal breakdown of some types of amino acids inside body cells. So immediately we've got the word amino acids, I'm thinking proteins. Figure 5, we've got some amino acids that have been eaten. Enzyme 1 then converts these into toxic substance P and ammonia. Enzyme 2 converts toxic substance P into a harmless product, and other enzymes convert ammonia into urea. A person with MSUD cannot make enzyme 2. Okay, so immediately we're thinking, okay, well, if they can't make enzyme 2, they're not going to be able to break down toxic substance P. So that could be an issue. So let's have a look at what the question is actually asking us. One of the final products shown in figure 5 is urea. Where in the human body are the reactions shown in figure 5 most likely to occur? Now, this question refers to the homeostasis topic in Unit 5. And what it's asking is, how does the body essentially process proteins, and how does it process nitrogen? You learn this when you learn about the kidney, because that is where urea is removed in urine. And one of the things we learn is that when proteins come into the body, they get digested into amino acids. They then get deaminated, so we remove the nitrogen out of those amino acids to make ammonia. That ammonia, as we can see in figure 5, is converted into urea, and that is then taken to the kidneys to be removed in your urine. So where do all these reactions take place? Well, deamination and conversion of ammonia to urea occurs in the liver. Okay, so a simple tick box question, but it's really stretching your higher knowledge. Easy marks if you know it. So just that little bit of extra focus on that revision can score you a few extra marks there. Lovely. So we then put that information into practice. So what it asks us is, scientists can analyze blood samples or urine samples to see if a person has MSUD. So immediately I'm thinking, okay, well, I'm not going to be caught out on these two. I've been told that they can do blood samples and urine samples. So let's keep that in mind. The test identifies high concentrations of toxic substance P, shown in figure 5. Explain why the blood of a person with MSUD will have a high concentration of toxic substance P. Use information from figure 5. Okay, so we know this is an explain question. So if it's an explain question, we need to be using biological knowledge. In other words, if it says explain, you need to use things that you've learned from the course and give a biological reason as to what's happening. Okay. So here it's saying, explain why the blood of a person with MSUD will have a high concentration of toxic substance P. Well, we've just been told that they do not have any enzyme 2. Okay, so if there is no enzyme 2, that means that they cannot break down toxic substance P. So we're now trying to think, okay, but why would they have a high concentration of toxic substance P in their blood? Well, if they can't break it down, that doesn't mean it's not made. Toxic P is still being made. So it's still being made as the person is still eating protein. And so that toxic substance is still being made. If it's still being made, it's still present. That toxic substance is being made in the liver, remember? So what we can then say is the toxic substance P, because it's being built up, is going to diffuse into the blood. And those are going to be our 3 marks. Okay. So 3 marks, we need to say 3 things. Here, no enzyme, so we can't break that substance P down. It's still being produced though, and so it produced in the liver and diffuses into the blood. Fantastic. Fantastic. Right, so moving on to the next part of the question. It says, explain why a person with MSUD must have a low protein diet. Now, this is a question that really tests some more of your knowledge behind the scenes, okay? So again, an explain question, and then again, 3 marks. So we need to be writing 3 things. Well, we can see that, or we can state the obvious, okay? So we've been told that it's all about amino acids in these previous questions, because that's how it starts. And we know proteins contain amino acids. So let's write that down. Start with, we can say proteins contain amino acids. Okay, so that can be our first point. Let's make that a little bigger. Fantastic. So proteins contain amino acids. If proteins contain amino acids, we need to keep the amount of protein low, or we need to keep the amount of amino acids low, and therefore we need to regulate their intake of protein. So we must keep certain amino acids low, and therefore have a low protein diet. But why is this? Why does a person need that? Well, they need it in order so that toxic substance P doesn't build up. So toxic substance P does not build up. And again, keep asking yourself why. One of the best tips for an exam question where it says explain is keep asking yourself why. If I've written it's important that toxic substance P does not build up, why is that important? Keep asking yourself that question. Get as much out as you can. So if toxic substance P does not build up, why is that important? So that it does not harm the body or cells or tissues or anything within the body, okay? So keep asking yourself, why is this the case? Why is what I'm writing important? And fluctuate or extend out that answer, okay? Fantastic, awesome. Hopefully everyone is following along nicely. Do let me know in the comments if there's anything you'd like me to explain more or go over. But we're just going to take a little second here just to recap what we've done. So we've looked over explain questions, we've looked over some tricky maths questions, we've looked over some tricky genetics questions. A Punnett square question always is one to remember. I can't remember the last time I've seen a paper without one. They're very common to be tested on, so make sure you can ask that question. In recent years, they've been making those Punnett squares 4 or 5 mark questions rather than 2 or 3. They're trying to get more out of you, so do be aware of that. Now, what we've got to do for the last 15-20 minutes of the lesson is to look at a few other areas that are really important. First of all, are describe questions. So these are very different to explain questions, and we just need to understand the difference. And then we will look at some evaluate questions and the dreaded 6-marker questions that come up every year to give you a little practice on those too. First of all, just a very quick question here, just a 2-marker to illustrate the importance of scribe questions that you find within these papers. So here a question says, a student investigated the effect of temperature on the time taken for different types of milk to decay. So we're looking at temperature and the decay of it. This is the method they used. They put cow milk in 6 test tubes, keep each test tube at different temperatures. They measured pH of the milk in each tube every day for 12 days. Record the number of days taken to reach pH 5. Repeat steps 1 to 4 with goat's milk and with almond milk. We can then look over here. So the students improved the investigation to produce valid results, and figure 1 shows those results. So you can see that we've got on the y-axis time taken to reach pH 5 in days, and on the x-axis temperature in degrees Celsius. We've got key, so we can see cow's milk, goat's milk, and almond's milk are just different solid or dashed lines. And our question is simply describe the effective temperature on the time taken for goat's milk to reach pH 5. So, and it also says here, use data from figure 1 to support your answer. Even if it doesn't say that, I would, just in case. It shows the examiner you know what you're doing. So anytime you're asked to describe a graph or a table, use information from that table. Describe questions. They want nothing about any biology, okay? Anything you've learned in the course, fantastic, but they're not for described questions. Don't go talking about how pH will denature enzymes that affect decay. Don't go and talk about how the enzymes will lose their shape and the active site won't fit the substrate. Don't give any explanation. All the described question is testing is whether you can just think scientifically about data that you are given, okay? So in this question, we're asked to describe the effective temperature on the time taken for goat's milk, so the solid line here, to reach pH 5. Well, we can see from this diagram, sorry, goat's milk is the dashed line, so this one going down here, the dashed line here. So goat's milk, we can see it decreases. What is the effective temperature? Well, as temperature rises, which we can see on the x-axis, the time taken to reach 5 degrees, to reach pH 5, sorry, reach pH 5, decreases. So we can see that as the temperature rises, the time taken to reach pH 5 decreases. We can even say from 11 days to 2 days. So I can put that in brackets, from 11 days to 2 days. We can then see more because it changes. That's part of what happens. And then we can see when it gets to this point here, it flattens out. Okay, so we can say at 15 degrees or above 15 degrees in temperature, the time taken to reach pH 5 stays the same. Okay, and those are your 2 marks. One for saying it decreases to start with, and another for saying it stays the same. You could even say it plateaus. Fantastic. So that is our describe question. No biology specifically, we're just describing the graph. Now also remember, you might be asked to draw a graph in your exam. We're not going to go into detail of how to do that in this live stream, but I do just want to use this graph here to illustrate the points. So remember, if you're drawing a graph, you need to use as much as graph paper as possible. The mark scheme usually says more than 50%. You need to include a label for your axes. You need to label them correctly. You need to include units. So in this case, in days on the y-axis, degrees Celsius on the x-axis. You need to plot your lines or your data points accurately. So if you're given a table that has this information in and asked it to plot it on the graph, you need to be accurate as to where you're plotting them. And then if you're asked to do a line of best fit, you need to make sure that line goes through as many points of your data as possible. Okay, so just remember, you might be asked to draw a graph in that exam. It's really a nice question though. If you follow those points, you can score a nice, easy 4 or 5 marks. So really good one to practice. Fantastic. So for the last 10 minutes, we are going to tackle one final question. We're going to look at a few more explain questions, do a bit more content from the higher tier, and we're also going to look at a 6 marker and some methodology. Fantastic. So final question for today. Let's have a look. So this one is about glucose and blood glucose concentrations. So here is our question. It is important to keep the blood glucose concentration within narrow limits. Person eats a meal containing lots of carbohydrate. This causes an increase in the person's blood glucose concentration. Explain how the hormone insulin and glucagon control the person's blood glucose concentration after the meal. So what we can write for this question, it's a 5-mark question, so we need to be writing 5 things. This is a big question, as you can imagine, but it doesn't have to be lots and lots of writing. Remember, you don't have to write for loads and loads and loads of lines. The mark schemes are very concise. They're just a few words. So you just need to be making the main points rather than waffling on in a little paragraph. Keep it to short, succinct points. So let's remind ourselves, how does insulin and glucagon work? Well, remember, insulin and glucagon are released from the pancreas. Insulin is released when blood glucose levels get too high, and it has the effect of lowering those glucose levels back down to normal. And glucagon works when the blood levels are too low, it raises them back up. I always remember that with the acronym GIRL. So glucagon here, G, raises blood glucose levels when they're too low, and insulin here lowers blood glucose levels when they're too high. Okay, so a good little acronym there to help you in the exam. So what can we write for this question? Well, explain how the hormones insulin and glucagon control a person's blood glucose concentration after the meal. Okay, well, the first thing that's going to happen is because they've eaten the meal, blood glucose levels are going to increase. Okay, so insulin will be secreted from the pancreas. So we're just going to think logically. If insulin is released, it's going to have an effect. It's going to want to lower the blood glucose levels. Now we can remember, how does insulin do that? Again, we're thinking more, we're thinking how. Why does all of this happen? Well, insulin will lower blood glucose levels, and it can do this by several different methods. So it can either cause the glucose to be converted to glycogen. Remember, glycogen is just a storage carbohydrate in the liver and the muscles of just lots of glucose attached to each other. So we'll lower glucose levels by causing glucose to move to be converted into glycogen. And also another method it can do that we learn is that it can be, insulin causes cells to uptake glucose, so that glucose comes out of the blood and into cells. therefore lowering the blood glucose concentration. And cause glucose to enter the cells and leave the bloodstream. What happens then? We need to talk about glucagon. So if we're lowering the blood glucose concentration, we get to a point where we probably go a little too low. So we then need to stimulate glucagon. So because blood sugar levels decrease to low, it causes glucagon secretion. And what does glucagon do? Well, we've just said glucagon will raise blood sugar levels. And again, we can explain why. We can say that it converts glycogen to glucose so that it can be released back into the bloodstream. Okay, so a good little bit of revision. A tricky topic, insulin and glucagon, a higher level topic if you talk about glucagon. So one there to remember. So moving on, we are going to tackle this question here. So metformin is a drug used for treating people who have type 2 diabetes. Scientists investigated the effect of metformin with 2 other drugs. Okay, so they're linking insulin, glucagon, and sugar with the diabetes topic. Scientists investigated the effect of metformin and 2 other drugs, A and B. The scientists wanted to see how the drugs affected the blood glucose concentrations of 220 people with type 2 diabetes. What did they do? They put 220 people into 5 groups, treat each group with a different drug or combination of drugs, and give each person a meal high in carbohydrates, and then measure the blood glucose concentration of each person 30 minutes after the meal, and again 3 hours after. Now, this is a scientific case study, if you will. It's an experiment that they've done. So we're asked to suggest 3 variables that the scientists should have controlled in the investigation. Again, something that comes up every year, and this time we're thinking about our scientific methodology. So variables, really important. We have independent variables. This is a thing that students still, even going into that last exam, still can't get to grips with. So independent variables, these are the things that you change, okay? So an independent variable is something you change. In this case, we're changing the drug to see which one has the best effect, okay? So the variable you change, for example, the drug. The dependent variable, this is the thing you measure, okay? The variable you measure. Now, in this case, the variable we're measuring is blood glucose. So blood glucose concentration. Control variables, which is what this question is asking about, are things we keep the same. So this is how we make an experiment fair. It's how we keep it in one way. So we're able to say 3 control variables. Now there can be literally anything. It's a fantastic question because you can write, literally there's 15 different things you could write for this question. So think about the experiment and think, what does it involve? And think, what do I need to keep the same? Now, importantly, I think the most important thing is we've got people in this experiment, and all people are different. So we need to keep the age of the participants the same, okay? We don't want it to have really young people and really old people because the effect might be different for different age groups. So age we want to keep the same. The height of the individuals and their mass needs to be the same. The number of males to females needs to be the same because they might have different reactions depending on male or female. Do the people have different severities of diabetes? How active are the individuals after their meal? If they're more active, they're going to use more glucose. If they're less active, they're going to lose less glucose. That will affect their concentrations. What types of meals are they eating? Is it the same meal? Does it have the same amount of carbohydrates? All of these things, okay? So take the method it's given you and dissect it to find things we need to keep the same. Often if you're looking at like practical experiments in the lab, it's often same volume of solutions, or same concentration of solutions, or same number of bacteria, depending on the investigation. Okay. Fantastic. The question then goes on to say, the scientists recorded their results as a mean value for each group. The scientists calculated the standard deviation for each group's results. Standard deviation is a measure of the spread of the individual results above or below the mean value. The scientists gave each group's results as mean plus or minus the standard deviation. The larger the standard deviation, the greater is the spread of results around the mean. Now, what that means is if you have hundreds of results, rating 1 through to 14, and you've got some that are 6, 7, 8, 9, 10, 4, whatever they might be, you're going to calculate a mean. So the mean will be kind of an average, and the standard deviation is the spread of data around that average. Okay, so a large standard deviation means we've got a large spread of data. A small standard deviation means that actually most of the points we collected are centered around that mean in the middle. We're asked here, which of the results is the most precise? A simple one-mark question, but that can be tricky if we don't understand the meaning of these words. Now remember, accuracy, that is about how close is the value to the true value. Precision is more about how precise is that value, meaning how within limits does it fall, okay? So what we want here is the value that has the lowest standard deviation. A precise value is one that doesn't deviate much from the mean. So because our plus or minus is the smallest, for this we have the most precise value. Most of the results are centered around our mean. Now, finally, we are going to tackle, for the last few minutes, a 6th marker, okay? Nothing to be scared of. This is a really tricky 6th marker here, but we're going to take our time in the last 5 minutes and just work this through. So here, Table 3 and Figure 9 show the scientists' results. So we've got the drug used. As I said, they did metformin A and B, and they also did combinations. So we've got metformin A and metformin B. We've got the number of people used in that trial, and we've got the mean blood glucose concentration 30 minutes after the meal in milligrams per 100 centimeters cubed, plus or minus the standard deviations again. In Figure 9, we've got a graph which shows us the mean percentage reduction in blood glucose concentration 3 hours after the meal. So how much has been reduced, how much has the blood glucose level been reduced by, and the drug used. Okay, so we can see here our bar graph, and we've also got our standard deviations shown. So what is our 6th marker question? In Table 3 and Figure 9, some standard deviations of results overlap. So we've got the table in the graph here again. Some standard deviations of results overlap. An overlap of standard deviations shows the difference between the means is not significant. In other words, because metformin and A and B all overlap, their standard deviations are overlapping, there is no difference in the effect of any of those drugs. If there is no overlap of standard deviations showing a significant difference, there is a significant difference between the means. For example, metformin with drug A does not overlap with any of the single combinations. Okay? So that shows this is better than any of the single combinations. A student looked at the scientist's method and the results in Table 3 and Figure 9. The student stated metformin works better when used with other drugs. Evaluate the student's statement. Now, if we're using evaluate questions, we need to make a judgment. We need to give an opinion, okay? This will be strongly linked and logically supported by sufficient range of reasons, and those reasons are going to come from the graph. Okay. So with questions, immediately start thinking for and against. So how does the information they're given support the student's statement? Well, first of all, metformin plus drug A gives a larger reduction in blood glucose levels than metformin alone. Okay, so metformin plus drug A gives a larger reduction than either drug alone. This is supported by the fact their standard deviations, which I'll abbreviate to SDs, do not overlap, as we can see on the graph. Metformin A here does not overlap with any of the drugs. So the statement is supported. If we look at metformin B, it also gives a larger reduction in blood glucose than metformin alone. Okay, so either drug combination, metformin A or metformin B, gives a larger reduction than either alone. Okay, so we can say the same about metformin plus drug B. We've got 2 points here, really. We've got one that metformin A gives a larger reduction than either drug alone, and that is supported by the fact that the standard deviations do not overlap. That would score 2 marks. We'll score a mark here by saying the same thing about metformin B. This shows a greater reduction than either of the drugs alone. This time, if we look at the standard deviations, we can see the lower standard deviation here, it overlaps with the standard deviation of metformin A and B all on their own. Now, what that means is that, yes, it causes a reduction in blood glucose levels, but it's not significant. It's good, but it's not something that it's not so good that we're really interested in it. Okay, so we can say, however, so this is an argument against their statement. However, metformin plus B's standard deviation overlaps with the metformin standard deviation, which means the difference is not significant. Okay, so we're not too worried about it. So we've got 4 marks there. What also comes from evaluate questions is the scientific methodology. Okay, so think pros when you think evaluate, think cons, and then think about the experiment as a whole. Okay, so what's wrong with this experiment? Well, immediately, we've only used, I think it was 220 people, it said in the previous part of the question. It's not a very large case study, not a very large amount of people. So we can say they need to use more people in their experiment. That's going to make it more valid. Okay, so use more people, and so we get an understanding of if it's for everyone. Additionally, the number of people in each group here, that is no good. It is no good that we use 25 people for drug B and then 65 people for metformin A plus A, because we're going to get a much better understanding if we use more people. We're going to have more data. So use the same number of people from each group. And additionally, we can say, because they're not doing these things, the data might not be representative or reliable, okay? And that's us to market. So anytime you see an evaluate question, think, what are the pros, what are the cons, and what about the experiment is good, or what, in more of the case, what is not so good. Fantastic, awesome. I really hope that has been helpful. If you have any questions, please feel free to leave them in the chat. Let me know whether there's any extra tips or questions that you have. But I shall leave this on the screen as the final thing. This is the top exam tips. So again, we're going to just be making sure that we use our command words. Be aware of how many marks are on offer. Again, the question won't trick you. It's going to give you lots of new information, but you have the skills to use them. We've seen lots of maths and practical questions, and again, as always, keep an eye on the time as you go through. Fantastic. Really hope this live stream has been useful. Any questions, I will be around. Please leave them in the comments. Thank you very much and good luck with your exams for the rest of the season.